Okay, so we're going to try this one more time. So protein sorting and degradation. So we've gone through replication of the DNA, the transcription to RNA, the translation to protein, the protein folding and sorting, the organism's structure and function. We'll be talking about the ubiquitin and ubiquitination, and protein degradation. Okay, so let's hop right into protein sorting. So all protein translation starts with free ribosomes. They can either finish with with a free ribosome, therefore proteins are located in the cytoplasm, or 2 finishes in the ER, the endoplasmic reticulum, therefore the proteins locate in the lumen of the ER. There's 2 basic forms of targeting pathways, post-translational targeting or co-translational targeting. Now, protein sorting is required to send finished proteins to particular cellular compartments. There's 2 basic forms of targeting pathways. There's post-translational targeting, which happens in the nucleus, the mitochondria, the chloroplast, or even the peroxisomes, or co-translational targeting, or secretory pathway, however you want to refer to it, which happens in the ER, the Golgi, the lysosomes, the plasma membranes, or the secreted proteins. Signal sequences are recognized by the signal recognition particle. So this SRP is a G protein, and it's a ribonuclear complex, and it binds to the ER signal sequence. And because the signal sequence is often cut off, methionine is not always the first in the final product due to cleavage at the cleavage site. So the SRP recognizes a charged amino acid, which you see here, and then a hydrophobic region, which is what you'll see here. So the charged amino acid is not always right at the beginning. Sometimes it can fall in between, but that's kind of the gist of it for what we have to learn. Okay, so the SRP targeting cell, what it does is it sees the mRNA with the ribosome complex. The SRP binds down on to the ribosomal complex, wherein it binds to the SRP receptor, the alpha part of the SRP receptor, which docks the ribosome onto the complex. Then when the SRP is bound, this opens the translocon, the GTP that is there on the alpha SR receptor and the SRP binding point, both are transferred into GDP, and then this binding opens the channel. The SRP is released, and it goes off and looks for other proteins to bind to the ER membrane. So, once the translocon is open, the protein translation continues. The signal peptidase cleaves off the signal sequence, which is this little bit here, which is obviously what the SRP was searching for in the first place. And then eventually the protein continues and finishes up being read and printed out. The RRF ribosomal releasing factor releases the ribosome, the translocon closes, and the protein is folded. Okay. So the various secretory pathways from the ER, it can go out to the membrane, out of the cell, or to vesicles. So as you can see here, you have the rough ER, which is where you have the binding sites. They're transported in transport vesicles into the cis Golgi, which are then smushed forward and pushed forward and smushed forward to the trans Golgi, where it can be either put into a secretory granule, or a protein inserted into the plasma membrane, or even into a lysosome as deemed necessary by the cell. Okay, so on that topic, in order to keep a cell working, it needs to remove incorrectly synthesized proteins, damaged proteins, cell cycle specific proteins, or other signaling proteins which are no longer necessary. So protein degradation, that's what's going on there. The 2 major protein degradation pathways are lysosomal, which is non-selective, or proteasome-mediated, which is selective. So let's go over lysosomal first. The lysosomes are acidic vesicles that contain about 50 different enzymes involved in degradation of proteins and other molecules. So proteases, or cathepsins, are the majority of what you'll see in lysosomes. They have a maximal activity in a lower pH environment, and they cleave the peptide bonds. Phosphatases removes the covalently bound phosphates. Nucleases cleave the DNA/RNA. lipases cleave lipid molecules, obviously lipase, lipid, nuclease, DNA, makes sense, through phospholipase C and other such lipases. And there is carbohydrate cleaving enzymes that remove covalently bound sugars from glycoproteins. Lysosomes can target damaged organelles in a process called autophagy, which is a self-defense system. It ruptures the inside of the cell, resulting in autolysis, also called apoptosis, or programmed cell death, which we talked about much earlier in the semester. Now, while lysosomes can also target these cells when the cell is in sort of a state of emergency and only needs specific functioning bits in order to survive, so certain organelles can be lysed in order to produce the remaining energy necessary for a short period of time, under a response stress condition, if you will. Okay, so the proteasome-mediated protein degradation is selective. So as we've learned, there's interphase, and then you have cyclins that control everything between that and the mitosis, and then the CDKs, again, controlling the interphases on the other end. Normally the cyclins have a higher amount, at certain points in the cell cycle, and lower points at others. However, under pathological conditions, you'll note here the line is thick all the way across, leading one to understand that the cyclin is high, which also causes the cell to be stuck in a mitotic phase. Okay, so proteasome-mediated proteasome-mediated protein degradation. So there's two major steps that we're going to be discussing. Is that the ubiquitin, a small 76 residue protein, is attached to the protein. Ubiquitin tagged proteins are recognized by the proteasome, which cleaves and degrades the tagged protein. Okay, so we have ubiquitin. It's attached to the target protein, proteasome. And so let's get into answering the questions of how ubiquitin is attached and how proteasomes cleave target proteins. Okay, so the ubiquitin has a C-terminus glycine, which attacks the amine of lysine. The amine is not alpha on this amino acid because it's side chain amine. So it targets the protein, which causes downstream effects, and you begin to see that the lysine will have ubiquitins bound to each one of the lysines. Often whole entire chains of ubiquitins are bound, so the proteasome recognizes the chain and then degrades the protein. Okay, so let's go into a little bit detail on protein ubiquitination. So say you have a free ubiquitin with the glycine end, lest we forget glycine end on the C-terminus, and some free ATP. So E1 will bind with ubiquitin. It's very generic, used for all sorts of things, all sorts of protein ubiquitinators, and it cleaves the ATP to release the pyrophosphate, creating ubiquitin with the AMP, which is E1 is a ubiquitin activating enzyme. So all the different ubiquitins, never mind, we'll get to that in a minute. Okay, so the E1 is cleaved off, or is put on, the AMP is taken off. The thioester bonding ubiquitin activation is caused by the UB to the E1 on the AMP point, which is this thioester, obviously by the S, ester bonding. So the E1 is transferred to E2, and the E2 ubiquitin is conjugating enzyme. So the E1 falls off, the E2 comes on, and you create this complex here. So then the E2 ubiquitin complex is found by E3, this big holdy molecule, hand molecule here, if you will, which brings the ubiquitin E2 together with the target protein with its free amine end. So the E3 transfers the ubiquitin of the protein into the ubiquitin ligase. So the E3 only recognizes a very specific E2, and it also only recognizes a specific target protein, in this case the glycine. So then the ubiquitin on the ester bond here that we see will bond to the amine bond here on the target protein, and then this is how the two are bound together, which is then targeted for proteasome-mediated degradation. Okay, so on to the proteasome itself. So it's a huge protease complex. It has a 19S cap, a 20S catalytic core, and another 19S cap on the other end. So the cap has three functions, which are to recognize the protein ubiquitin, cleave the ubiquitin protein off, it unfolds the protein to be non-3D or peptide, and sends the rest of the protein into the core, where the protein degradation occurs. So total 26S proteasome in this sample here. Okay, so proteasome-mediated protein degradation. So the ubiquitin protein is found, ubiquitinated protein. The cap recognizes the proteasome goes in, it releases the ubiquitin, and it causes peptide fragments, which go through proteolysis, which fall down into their amino acids, which can be left intact for biosynthesis or into amino groups, which can be dispersed by the urea cycle, or can spread into the carbon skeletons to glucose or glycogen synthesis, into fatty acid synthesis, or cellular respiration. So a whole lot of different ways that the proteasome degradation can lead in product-wise. Okay, so the proteasome-mediated protein degradation physiological significance is that something like Taxol, which can arrest cells in mitosis, which leads to cell death. So it doesn't inhibit the proteasome directly, but it disrupts the spindle structure, which inhibits the DNA synthesis later down the road. Okay, that's a real quick and dirty rundown of everything that we were talking about there. I hope you had fun with that. I know I sure did. And that's all we've got for protein sorting and degradation. You should now be an expert on DNA transcription, RNA translation, protein folding, sorting, holy cow. I cannot say words here. Hold on. Organism structure and function, and protein degradation. And that's all I've got for tonight. Have a good day.